Kinsa mo present na to, Rab?
Ako laban. Sige, wait.
Sige, sige.
Kinsay mo introduce sa inyong topic. I want to see the objectives of the paper, ha? Someone should introduce the topic and then what are the objectives of the paper or of the article, followed by the content. Because what I have given you is a review paper, right?
Wait. So, good morning, everyone. So today we are to present the article Adaptations of Archaeal and Bacterial Membranes to Variations in Temperature, pH, and Pressure. So, wala nang nabutang ng objectives. So the objectives of the paper involves an overview, the description, to describe the adaptations.
Introduce the topic first before you. Kung sa may una, sa paper presentation, what is first? Diba? Title, followed by the introduction. And then gap or the problem, statement of the problem, followed by the objectives, the significance of the study. Diba? That's how you present a paper.
Yes, I guess. Wala, may kagabaaran, ma'am.
Ano nga? Ang saan man nga? How do you introduce a paper?
Sulti na lang nako, ma'am.
Sige.
Yes.
Gaya nga, dahil mas content, wala dahil ninyo gyapil ang introduction, di ba na yung kuya, apil ang intro o ang summary, conclusion.
Yes, ma'am. Dagdag.
Then let me see your PowerPoint presentation, Osa. Sige. Okay. Imuha ni tanan Montegreho?
Ma'am?
Including pH? Imuha na?
Ang pH ay ako, ma'am. Katong
Okay.
temperature is Jezreel, ang pressure kay Janelle.
Inyong gi-merge ang PowerPoint presentation?
Yes, pero na may copy tag sa ma'am.
Okay. Sige.
Balik. So, ako. So, again, so the adaptations of archaeal and bacterial membranes to variations in temperature, pH, and pressure. Bacterial and archaeal lipids are inherently different from each other, as so evident by the so-called lipid divide, wherein bacteria and eukarya have phosphatidic acid, while archaea have archaetidic acid. Now, despite this being true, There are instances wherein either domains show similar responses when exposed or when subjected to similar environmental conditions such as in the title, temperature, pH, and pressure. So the objectives of this article is to give an overview on the adaptations of the core lipids in bacterial and archaeal cells in response to changes in physico-chemical conditions. Then it also gives a comparative analysis of the reported growth ranges of the most robust extremophiles to date, as well as to disclose the growth boundaries of the bacterial and archaeal domains. Then there is also a discussion on the boundaries in relation to the encountered lipid compositions and their adaptation to the archaea, in their adaptation in the archaea and bacteria. So the lipid divide, as I have mentioned, is the observable difference that is evident in the plasma membranes of archaea and bacteria. So if you're going to look at the figure, so you'd see that. So the first is ang parsa niya. So the gray hydrocarbon chains are represented by fatty acids in bacteria and isoprenoid chains in archaea. Sorry. So you could see na in bacteria there are two types too. So you have these straight chains here na it's a na ang iyahang kuan is fatty acids, kani iyahang hydrocarbon chains is made of fatty acid. Compare that to the isoprenoid chains of
One hydrocarbon chain is made up of fatty acids. What's the difference between the hydrocarbon chain and fatty acid?
pardon ma'am.
What's the difference between a hydrocarbon chain and fatty acid?
Hydrocarbon chains are inherently carbons bonded with hydrogen. So ang main difference nila is that fatty acids can be unsaturated or saturated, whereas hydrocarbon chains are purely saturated.
Are you sure?
No.
Can we check on that, Mr. Montegrejo? Continue.
Yes. Then the red portion of the diagram is that in bacteria,
Di ba hindi
ester
naman
bonds,
organic? Ano naman with organic, di ba?
ma'am?
I suppose. You're done with organic chemistry now?
Yes, we are.
Actually, fatty acids are just hydrocarbon chains. Because fatty acid basically is composed of a chain of carbon
Hydrocarbon
bonded
chain,
to hydrogen. Yes. And hydrocarbon chains is not limited only to
unsaturated.
saturated or unsaturated ba na siya. Because hydrocarbon chains may also be connected to other groups. Pwede siya connected to cyclic compounds, pwede po siya connected to isoprenes, okay? So these are limited to carbon and hydrogen only, but there can also be other chemicals or other compounds that can be connected to hydrocarbon chain. Therefore, changing, of course, the conformation of the molecule. This is also responsible for the variety of or for the fluidity of the membrane. Okay? But basically, these hydrocarbon chains comprise the fatty acids found in the the plasma membrane. Okay?
Yeah. Yes. Oh yeah, sakto. So then sa red na portion, the red portion indicates that in bacteria, the hydrocarbon chains are bonded via ester bonds, while to a glycerol backbone, while in archaea you have ether bonds, by ether bonds. Then the yellow portion is the head group, which is In bacteria is represented by glycerol 3 phosphate and archaea glycerol 1 phosphate. Yes, so bacteria and archaea evolve domain-specific adaptation mechanisms to respond to different physicochemical conditions because of chemical differences. So as evident in the figures, you could see that despite being different, they function similarly to facilitate protection of the interior of the cell and as well as the entry and exit of materials of molecules that the cell can metabolize. So a change in lipid composition is regarded as a stress response. So despite this being established nga there are despite being the lipids or the four types of lipids that are involved in that are present in the cell, there are instances when the cell or the organism changes its lipid composition as a form of coping in an environmental condition. And we'd call that a stress response. So low temperature adaptation.
Remember,
Just
these are divided, excuse me, Mr. Montegrejo, these
yes,
adaptations are divided into two, long-term and short-term, right?
yes,
Can you discuss each of those mechanisms?
yes. Um, the long term, adaptation of cell physiology to physicochemical conditions occurs at two levels. So we have the long term and the short term. The long term is the genome evolution, which defines the range within which a cell can survive. So it is defined ano, the presence of the lipids is defined by the range at which the organism is present. So for instance, if a typical bacteria, for instance, is an acidophile, so the components of its cell membrane is adapted or is configured such that it is efficient whenever in acidic environments. So that involves uh, the presence of the presence of membranes that are not easily impermeable to protons such as GDGTs and tetraethers. So inana. Whereas short-term refers to the reversible regulation of gene expression and enzyme activity to achieve optimal functionality. So this ang short term is those adaptations na makita whenever an organism experiences stress sa iyang environment. So for instance, if a neutrophile is exposed to a slightly acidic environment, so na siya mga coping mechanisms, which may sometimes resemble that of those nga mga obligate nga acidophiles. So, inana.
Okay. What about homeostatic, what is it? What about homoviscous adaptation? What does it mean? Can you describe also T optimal, T maximum, T minimum before we proceed with the different adaptations so that as you go on with your discussion, as you mentioned with these things, it's clearly defined first okay, para dulo-diretso. Because you
So,
will be using
homoviscous
this term.
adaptation is the adaptation, ma'am?
Go on.
Okay. So homoviscous adaptation is the adaptation of the cell membrane lipid composition to keep the adequate membrane fluidity or permeability. So later on, i-discuss ng what is the importance of membrane fluidity. And ang iyang main is that the fluidity of the membrane should remain relatively constant at various temperatures, regardless at various temperatures or any environmental conditions, because the fluidity of the membrane is an important factor in cellular metabolism. So, dapat siyang ma-maintain, and that is the reason why katong na ay mga short-term adaptations as well as long-term adaptations. So, tayo naapod ningon siya nga physiological membrane adaptation, or this kind of adaptation is regarded as native phenotype, which is the lipid composition that enables a species to thrive at a particular challenging habitat. So it's like akin to what a long-term adaptation is. Dayon, alternatively, there is also the stress response, which, as I have said, is the changes in the lipid composition when conditions of the natural habitats change. Yeah.
What about native phenotype? Is it long-term or short-term adaptation?
Yes, ma'am. Ang native phenotype is the phenotype na makita that is observable in an organism when it is exposed in its normal environment,
Okay, my
normal or
question
natural
again,
environment.
is it long-term or short-term?
It's long term.
Okay.
Yes. So I am to give the platform now to Mr. Banagodos for his report. Jess, do the honors.
Okay. Wait. Nakakita mo.
Kita ra.
So, low temperature adaptation in bacteria. Now, sa low temperature bacteria, which is regarded as cyclophile saprophilic bacteria, bacteria
Bacteria.
bacteria must retain the fluidity of their membranes to allow metabolic functions, as which special membrane proteins on liquid crystalline phase. Now, this is the reason why bacteria must retain fluidity of their membrane because of the metabolic functions. But based on the physiochemical conditions, the adaptations of the membrane may vary within temperature. Below the transition midpoint of the lipids, below the transition midpoints, lipids may froze, and above the transition midpoint, the phospholipids hydrocarbon chains are motile. So, Marshall,
Is Mr. Banagodos, is the transition midpoint the same for all species of microorganisms?
ma'am,
Is the transition midpoint the same for all species of microorganisms?
dili.
Okay. So what makes it different sa psychophilic and thermophilic microorganisms?
Ang ilahang optimal growth nila, either sa naman to'y mo ato niya atong graph nila sa atong minima o maxima nila. Ato.
Okay, last question before you proceed, Mr. Banagodos. Is the transitional midpoint the same with the temperature required for optimal growth of microorganisms?
Ang sa ma'am, optimal growth niya,
Ah, the same ba siya sa transitional midpoint?
not necessarily na same temperature niya kay ang transitional midpoint below minima kay below 15 degrees Celsius man ang katong above TM kay 7 kay greater than 75 degrees.
It's not the same for all microorganisms, di ba? Mo imong ingon ganina.
Dili gali.
And so,
Naiuban nga
correct. It's not necessarily it does not necessarily mean nga if mauna iyahang transitional midpoint, then that is the optimal growth.
oo.
Yeah, but what's the explanation?
Unsa explanation nganong dili
Yeah,
mo
not necessarily the same.
Kay dili man sila pareha po. Based sa kuan man siya ma'am, sana species or situation specific man ang adaptation sa membrane gland.
But remember, a transitional midpoint is only applicable to cell membrane. Whereas ang iyang optimal growth, you have to consider the metabolic activities that occur as well in the cell. Which is the reason why it doesn't necessarily mean if kana ang iyang nga transitional midpoint sa iyang nga membrane, mao po na iyang nga requirement for optimal growth, because we also have to consider the overall activity of the cell. Okay? Continue.
So the molecular mechanisms are directed at increasing the fluidity by forming a more disordered gel phase, and the fluidity of the bacterial membranes is dependent on the membrane's phase transition temperature, which in other terms transition midpoint. So these are the documented stress responses from low temperature adaptations of bacteria. Una, unsaturated fatty acids. No, unsaturated fatty acids are the most prevalent cold stress modification in bacteria. Pwede siyang alter siya cis-unsaturated and trans-unsaturated. Sa study na ilang sa paper na ilang gi-discuss dire na ilang gi-ingon nga as temperature increases, the conformation of cis to trans is relevant. So from cis to trans, must prefer ang trans unsaturated fatty acids in higher temperature than of cis unsaturated fatty acids. And I'm sorry. So
Go on.
So short chain fatty acids. Now, sa short chain fatty acids branch chain fatty acids kay dili silang ingon nga cold stress adaptation sa bacteria. Kay ang sa short chain fatty acids can only be implemented sa growing cells. So ang sa branch chain fatty acids kay ang BCFA, branch chain fatty acid response, is species or situation specific. So katong ilang so you read the paper na ilang experiment tong sa thermophilic bacteria na ilang sa tawagan na
The subject,
ilang subject sa low temperature niya ang ilang kuan dito kay ang pag subject nila is na a decrease of branch chain fatty acids sila nakita. So dili gaingon ang branch chain fatty acids kay stress adaptation in cold temperature in bacteria. Sunod
cold
kay
or low?
Low temperature adaptation in
Anyway,
carotenoids.
it's the same.
Kali. Carotenoids, which these pigments adapt a so-called membrane spanning orientation by inserting their hydrophilic groups at opposite sides of the polar regions. Because of these carotenoids, because of the membrane spanning orientation, fluctuation in fluidity in gel phase and liquid crystalline phase, carotenoids are said to balance fluidizing effect. So, ang carotenoids sa gel phase increase fluidity siya, and liquid crystalline phase decrease fluidity if mo insert ng carotenoids. Now, kay glycolipids and uncommon polar bonds. Sa
Sige, go on
sa glycolipids dili kayo siya kay gi-discuss sa paper, but it would be I would assume that its role would be to help stabilize membrane structure, and also that conferred membrane flow. It could confer membrane fluidity after exposure to cold temperatures.
Okay, what is
So
common, Mr. Managodos, before we continue? Can you go back to the previous slide? All right. So what is common to these molecules? Unsaturated fatty acids, SC, FA, carotenoids is common to these molecules
on
in
psychom,
terms of their molecular structure.
ang unsaturated or short chain fatty
The
acid
number unsaturated fatty acids contain or do
so
not contain double bonds?
contain double bonds.
Okay. What about branched chain fatty acids? Although they are not important in microorganisms, especially bacterial organisms that that are found in cold temperature or low temperature habitats, but there is common to these molecules. And what is that? In terms of their molecular structure,
Sa branched chain fatty acids also.
no, the four, including lipolipids.
Um,
By the way, you can ask questions to your classmates while discussing. Then let's ask your classmates.
both.
Or sa mga atong pangtanon. Miss Tubil. Miss Tubil?
Ng uncommon nila, ma'am.
Yes.
Kada ang glycolipids, ma'am.
More sure? I
Ay,
mean, in the molecular structure, the overall molecular structure, do you have a figure of these molecules with you, Mr. Banagodos?
Wala na ko na-appeal mo. Na, ako ang tanggi appeal pero wala na ko nabutang sa PowerPoint.
Okay, anyway, what do you think is common? Remember what happens if the temperature is very low? What happens to the membrane?
Muka.
No?
Ma, mura siya, mga to siya.
Ano sa
Gel. Sa bacteria? Ato gel phase.
What is true to all membranes, di ba? Mahimo siya mo unsa?
Mga to siya gel phase.
Alright, and so, in order to prevent that from happening,
Increase ang fluidity.
okay. So, in order to increase the fluidity, there is presence of these molecules.
Ako, kali.
Oh, so what is the difference with these molecules with other molecules na makita nimo sa mesophilic microorganisms?
Mm,
Right? Basically, these molecules function for spanning the plasma membrane, right? which is responsible for maintaining the fluid.
fluid.
Okay, maintaining the fluidity of the membrane. So if
Ano
you
yun?
are going to look at the molecular structure of these compounds, these are actually bigger and bulkier. Okay, mas bulky siya compared to other molecules. Aside from that, you are also going to compare the conformation unity and there is naay angle ang iyang mga fatty acids in order to prevent compact uh, ma-prevent ang pag-compact sa plastic. Okay? So that's the main difference.
Yes.
Go on. Continue.
So, as you can see na katong sa paper niya, ilang gibutang ang bacterial growth temperature optima versus percentage of branched chain fatty acids. So
Okay.
as temperature drops and grows either way, as you can see that and the temperature relatively that is relatively lower, gamay ang makita na branched chain fatty acids niya as compared to that temperature nga pagpataas. So muna ilang giingon nga dili jud dili stress response sa lower temperature ang BCFA, which is branched chain fatty acids. So there is phase transition in lipid bilayer. Okay, muna ilang giikuan nga above transition temperature and below transition temperature, which is muna conformity sa membrane kung na siya above transition temperature and below transition temperature, which is ang sa below transition temperature packing order siya. So compact siya nga pagka sa membrane niya as sa above transition temperature, which is either nga free floating kuan siya dili kay siya compact, which is because of the higher temperature. Marshall, kuan analogous siya sa kuan oil. Kanang if imong subject ang oil ninyo dira sa balay o lower temperature mo kahi siya. O imong subject ninyo siya sa higher temperature, which is mo liquid form siya. Marshall pareha sa Japan siya diri sa atong discuss.
Not just oil. I think it's applicable to almost all molecules.
Ah,
Remember, if ang water, if you subject it to lower temperature,
gahi.
diba? Not just
Yes.
oil, but almost all molecules, except for those molecules that have strong molecular bonds or chemical bonds found between molecules. Naka-resist siya sa compaction, but this concept is actually applicable to almost all molecules.
So sunod kay low temperature adaptation and archaea. So bacterial saprophytes outnumber archaea in diversity and dominate sub-freezing ecosystems, which katong ang archaea kay duha ka confirmed species na saprophytes, which is kaning na diri. Yeah, dili siya wala kayo study sa psychophilic archaea karon na nagi-publish. So wala kayo ang ilang gihimo na para maka-produce results kay ilang subject ang thermophilic nga archaea to lower temperature. Mura na ilang gibuhat sa uban. So muna ang pareha sa Japan sa archaea, ang archaea na ay mechanisms of adaptations from low temperature, which is una kay unsaturated ethers and sunod kay isoprene hydroxylation, which works by shortening the core of the lipid, and tetraether diether ratio and number of pentacycli. In tetraether diether ratio, the own experiment given was that katong thermophiles na yung subject sa low temperature below growth below optima, and the result increases the increase of diether and decrease of GDGT. So mas increase ang diether instead sa tetraether, and same as tetraether diether ratio, the number of pentacyclic is observed in hyperthermophiles. So nonetheless, the function believes to stabilize chain packaging and to decrease membrane permeability. So muna ilang ang ilang nakita ang adaptation sa low temperature in archaea. Muna katong graphs nga gikuan nga transition minima og optima og max sa between archaea and bacteria. So as you can see nga ang mas na-dominate ang sa bacteria kaysa sa archaea, instead nga dili siya makalapas kayo sa negative 2 degrees Celsius sa iyahang.
Aside from those, unsa pa imong ma-observe in between the two figures?
Wala kayo subject matter ang sa archaea, which is gamay ra kayo ang ilang gamay ra ang ilang gi-studyhan.
The species?
Yeah.
The number of the species in archaea that are used in archaea is lesser compared to that of bacteria. And aside from that, some of these species were actually thermophiles subject to lower temperatures. So what do you think is the change in the membrane composition of these thermophiles or thermophilic archaea, long term or short term?
Ang sa thermophiles? Short
Yes.
term
Okay.
is sunod kay ang high temperature adaptation bacteria, which is presented by Janelle Domanglas. Jan. Wala na question about sa low temperature.
Hi, people listening to Mr. Banagondos, do you have any questions before we continue? Ikay mangutanag question nila Banagondos, basig ga-discuss ka diyan, nangatulog na ning uban. Are you listening? Guys, kay off ra ba na inyong camera? May kabalog kaligad ba mo diha gaunsa mo? Can you ask question na lang? Can you ask question to your classmates?
Ako?
Yes, I don't know if they are listening, just to check if these people are still with us.
Ay kana yan sa kanina last paper. So ngano daw ang ngano ang metal branch phytanils kay nganong dili kasi tawag.
So, manjud ba nagudos?
Di ko ka formulate to question ani lagi.
Unsa man ngano ang methyl branches? Siyempre
Kay-
pasabot. Kay
nganong gamay ang metal branch sa bacteria compared to that of the archaea. O unsay functions na ngano ang mas daghan ang metal branch sa archaea.
okay, someone answer the question of Mr. Banagudos. Isa tiwa, natawag na nakustubil, no? Tupas? Tupas?
Yes, ma'am.
Hello? Hmm.
Hello.
Sige.
Um, sa Archaea, um, kuan jud siya, this is the imperative feature jud siya sa archaeal mga sa Archaea, ang methyl branched hydrocarbon chains and um okay.
Does that answer your question, Mr. Banagudos?
I
Uh,
have additional. Wait.
Sige. Ano po additional
Um,
statement?
ang katumanggo sa bacteria manggod naa sila methyl branched lipids but compared sa archaea mas daghan sa archaea bali gamay lang sa bacteria. So less sa bacteria, however sa archaea daghan, and then ang when it comes to mesophile, that's why effective, I mean, um kaning useful ang methyl branched hydrocarbon chains sa mesophiles. It's compared sa thermophiles kay kuan man gud kaning methyl branched hydrocarbon chains sa thermophiles disadvantage ni para nila.
Does that answer the question, Mr. Banagudos?
pwede naman.
By the way, Miss Tupas, this is kuan ha, outside sa atong discussion. I'm wondering, wala lagi si Abril. Kamu raman laging irengeng. Wala siya ni-enroll karon ni Sen?
Kuan ma'am kaning nahitabo man gud overload na siya. Dayon anak ko niya nga pwede nimo i-drop na lang ang imong minors. Ana siya di pwede kay ma-close naman daw ma'am.
Okay.
Oh, so ulahi
Dapat
jud
mo
siya.
minors na bilin? Dapat mo
Ha?
minors na bilin?
Unsa ma'am?
Dapat mo minors na bilin?
Siya.
Dapat ba mo minors? Siya na
Siya.
lang.
Maulahi jud siya.
Okay.
Gani ma'am
So,
sayang
dili pa siya
kaayo.
sa kadungan ninyo o practice teacher if in case
Yung
ana
second sem
practice teacher
dili
mo
next year pa.
maluyang Abril.
Mao gani
Sige,
ma'am.
sige.
Maluwi ko. Sige ma'am.
Be, kasi pa continue. Dapat I will still have 10 minutes.
Yes, si Katranel namang last naman.
Ano, di sa man ninyo pagbahin. Mo na to.
Ay, ikaw dyan, ako represent or ikaw?
Awa dahil mo gabahin? Daan?
Gabahin man.
No.
Hello?
Hmm. Sige, Miss Dumanglas.
Hello, nakita ra guys. Hello?
Yes, dyan nakikita.
I can hear you.
Okay. Um, high temperature adaptation in bacteria. When the temperature rises beyond the optimum, the liquid crystalline phase, lipids adopt a fluid phase and ultimately also a non-lamellar phase, like the cubic and hexagonal structure. They become disordered. So the microbes at high temperature endures increasing fluidity and permeability that in maintaining the liquid crystalline phase becomes disordered. The lipid adopt from a fluid phase and non-lamellar phase, such as cubic and hexagonal structure in which these structures are ordered and less packed. Thus, the bacteria must adopt mechanisms that stiffen their membranes as well as to promote order in this hydrocarbon chain. So, the mechanisms that the mesophilic and thermophilic bacteria mainly adjust fluidity and by increasing its branch chain fatty acids, saturated fatty acids, long chain fatty acids, and um, polar co- uh, uh, carotenoid content. In the branch chain fatty acids, high amounts of branched chain fatty acids at highest temperature, sometimes 100% of fatty acids, and composed of iso branched chain fatty acids. Um, branched chain fatty acids packs tightly to form rigid, high melting point extended structures and reduce phase transition temperature of membrane phospholipids. In the saturated chain fatty acids, are saturated with hydrogen since single bonds increase the number of hydrogens of each carbon. In the long chain fatty acid, tet- tetraether uh, ester tetraester lipids are present and are able to withstand up to 80 degrees Celsius but no greater. And lastly, the polar carotenoid content. The decreased membrane fluidity in the liquid crystalline phase, but increase in fluidity in gel phase. Okay. Um,
Miss Dumanglas,
yes.
I don't know if it's true to all, but your voice is somewhat hinay.
Um,
Is it the same for you guys as well?
can I?
Nicole?
Hello.
Yes, come on. That's better.
Oh, wait. Asa ang
I think
dili?
you can speak near to your mic. Okay na. I
Okay.
think you can speak near to your microphone if you're using
Yes, ma'am. Okay. Asa dapat itong like hinay kaayo kong tingog.
those four molecules.
Okay. Uh, the first one is the branch chain fatty acids, in which high amounts of branched chain fatty acid at highest temperature, sometimes 100% of the fatty acids, and it is composed of iso branched chain fatty acid or like okay branched chain fatty acid packs tightly to form rigid, high melting point extended structures and reduce phase transitions temperature of membrane phospholipids. In the saturated fatty acid, these are saturated with hydrogen since single bonds increase the number of hydrogens of each carbon. In the long chain fatty acids, the tetra kaan- e- ester lipids are present and are able to withstand up to 80 degree Celsius but no greater. And lastly, for the polar carotenoid content, the decreased membrane fluidity in the liquid crystalline phase, but increased fluidity in gel phase. So the next is the high temperature adaptation in archaea. For thermophiles, membrane spanning tetraether are the most abundant and frequently the only core lipid. So the tetraether by nature are resistant against heat, and thus increasing temperatures also means increasing tetraether and diether ratio. The tetraether lipids form monolayers that are highly stable due to the restricted motility of the hydrocarbon chains.
Excuse me, Miss Dumanglas.
Yes, ma'am.
Before we end, because it's almost 10, you have a class at 10 a.m., right?
Yes, ma'am.
Okay, so you've mentioned that there is an increased ratio of tetraether to diether in archaeal cell structure. Now, how is it different with what was discussed by Mr. Banagudos a while ago in terms of the ratio and why?
What's up? What's up? Um,
Not really see you, but uh, we'll conduct our meeting na lang tomorrow. We will continue with your article presentation tomorrow afternoon, 1 p.m. Okay? Thank you for your time. Stay safe. Bye. You can leave
Thank you,
now.
ma'am. Bye.
Thank you, ma'am.
Okay.
Hi, ma'am.
Bye.